All right. Well, welcome everyone to our 28th annual Tri-State Greenhouse IPM Workshops. I'm Cheryl Sullivan from UVM, co-hosting this workshop with Margaret Skinner in collaboration with other key organizers listed on the slide along the right. So these events would not happen without the strong network we have among growers, extension, state ag personnel, researchers, and other industry representatives, and funding agencies that help to support a lot of our work, like the USDA and the American Floral Endowment and many, many others that you'll see recognized on many of the slides that we have today. So thank you all for the support, and to all of you for coming here today to this workshop. So just a few details. This presentation is being recorded and will be sent directly to you. The recordings will be after the event, probably early next week because it takes time to process. Please make sure your microphones are muted. Ask questions by entering them in the chat unless we ask otherwise. And we're offering 2 pesticide credits for today and 2 more next week, but you need to stay for the entire meeting and take part in the poll questions that we're going to ask. And if you haven't already contacted me in that form that was sent out in the email requesting credits, please do so by typing your name and license number into the chat. So for all you New York applicators, please verify that you are here by putting your name and license number into the chat. New York specifically asks for that, as well as other items. So it's also recommended that you leave your cameras on so we can kind of make sure that you're still here so you can get some credit. So today, our first speakers are Mark Ray and Nick Skinner from the Icahn School of Medicine at Mount Sinai, New York, and they'll be discussing the use of UVC for powdery mildew. And I'm going to stop sharing, and Mark, you can take it away. Thank you.
Okay, I want to, you'll notice I changed the title slightly because we've done more than work in powdery mildew. And hopefully I realize that you folks are more in the controlled agriculture field, but I think the principles remain the same, obviously to be adapted to greenhouse applications and high tunnels. I want to acknowledge Nick Skinner. Nick's been with us since he was a student back in the early 2000s. He's now flying drones for NIPA, which is the state utility looking at maintenance. He's a pilot and said he couldn't resist the opportunity to do something new. Otherwise, he'd be the person today presenting. I also want to acknowledge Margaret and Bruce Parker, who we worked with a few years ago when we were at Rensselaer Polytechnic Institute. And so thank you for the invitation, and also Margaret for reaching out to I know that the New Yorkers are foreign to your group, but thank you for making that possible. I want to say a little bit about us because some of you may wonder, what does the School of Medicine have to do with plant health? This is part of a much larger view of health, if you will. And there are things, for example, I don't know how many people know about white nose syndrome in bats, but they are a fungus that has been quite lethal to bats. And many of the farmers have now started using more insecticides. And as a result, for example, infant mortality in those areas have increased. So this is an interconnected world. Physics is the same everywhere. And so we try to make that bridge between the physics of light and optical radiation into implications with regard specifically at health is what we're focused on right now. But we are involved in a number of areas like transportation, headlight design, energy efficiency, looking at that's part of the stuff Nick's doing now. And we have a photometric facility which actually quantifies light and optical radiation. And if I just make a snotty sidebar, one of the things I see in the literature across fields is very few people really accurately characterize light or ultraviolet radiation. And that's one of the strengths, I think, if we're really going to make progress, is being able to relate the physical world to these biological outputs. And so that's sort of the underlying theme that we've tried to carry from the time we were at RPI to today. Now, the punchline of this, and the reason why we're doing it, as I mentioned or alluded to, is that ultraviolet is an underutilized area in horticulture and agriculture, in my opinion. And if you look at the market from the manufacturers of light sources, they too do not see a large market for this, and yet it has these distinct advantages. It can be adapted to a variety of crops. It leaves no residue. Workers can enter an area immediately after the application, thereby reducing fungicide exposure risk. It can be applied to any weather, and it's cost effective. Starting from scratch, we have a DIY drawings that turn out to be, even though you have to put that into a new apparatus, we can show that it is cost effective by itself in 3 years, and in combination with things like other pesticides can be a payback of a little over 1 year. So let's be clear what we're talking about. The way we consider optical radiation or light ranges from about 700 nanometers, the red end of the spectrum, all the way down to 200 nanometers, just before it becomes an ionizing radiation, which I think is one of the areas we're staying away from. I just want to make the point that solar radiation includes everything but UVC. The atmosphere filters out all that, and UVA is the one that gives you sunburn. UVB is the one that provides vitamin D production through the skin. So this is sort of our platform, but what we're going to talk today is mainly about UVC, which is not available in sunlight. So the fundamental consideration when you are talking about an intervention of ultraviolet into controlling a plant pathogen is dose. Dose is how much of the UVC you're going to need in time to reduce, for example, a particular bacterium or fungi. And dose is the way we want to think about it, and that's measured in joules per meter squared. So it is composed of 3 key elements. One is the irradiance, that is just the watts per square meter. These are radiant watts, not electrical plug-in watts. How long you do it, and then we have efficiency, which is a unitless dimension, which simply talks about how efficient is that particular irradiance over time going to be for mitigating that particular pathogen. So dose, in simple terms, is irradiance times time times efficiency, or how much light for how long, and what are we talking about we're trying to kill. So Margaret will later talk about the difference in entomology. Thrips are pretty hardy creatures when it comes to UV. They would need then more dose to be able to be as effective as it might be for, say, powdery mildew. So visually, dose, it can be thought of as a reciprocal relationship between the amount and the time, the duration that you're doing. So you can, if you're driving a tractor, you can put a lot of radiation on the plant for a very short time, or you can reduce the amount of light sources you have, but drive the tractor slower, and you will get the same dose. Now, the efficiency where we have been primarily focused is in nucleic acid, both RNA and DNA, RNA for viruses, DNA for fungi and bacteria. And what we're doing is we're breaking down the bases, the fundamental basis that require replication in a pathogen, say powdery mildew. Now, one of the things that people always ask, and I think it's an important one, is that, well, we build resistance to chemistry. Recent studies showed that with UV breaking down DNA, it was after, I think it was 24 generations, there was no adaptation to this particular intervention. So this has also the added strength that once you remove its ability to reproduce, you probably don't have the wherewithal to do the replication. Now, this graph on the left, now that's wavelength. Remember, 200 is the beginning of UVC to 300, which is about UVB. This is what's called the spectral sensitivity of nucleic acid to optical radiation. And what you see is the peak sensitivity, meaning you require less optical radiation, a lower dose to achieve the same effect. So that whole curve is equal effectiveness, but varying as a function of wavelength. And what you see is the peak sensitivity, the least dose that you need to reach a criterion kill would be around 462 or 465 nanometers. Now, that's not the only mechanism, and we'll talk a little bit more about this, but there are also what are called chromophores that are what we call endogenous or inside the cellular existence of a fungus or a bacterium. And they will also absorb radiation. Now, they are not exclusively absorbing ultraviolet. Some actually, some of the work we're doing looks at blue light, which is visible. Also does things, for example, to help control the immune system in skin, human skin. So all these cells have different, what we call chromophores, that absorb different regions of the spectrum, have different efficiency that would then be also lethal to the pathogen. Now, honestly, we don't know how big a play these are in the applications we have. Our target has been, because of the efficiency, breaking down DNA, but undoubtedly some of these other chromophores are going to also be effective probably at breaking down the biological cycles that you're going to find in any animal or virus. So what is produced is what's called reactive oxygen species. So think of hydrogen peroxide as one of the common ones, and that is the thing that then breaks down some of the cellular functions. So again, I don't know exactly on our applications how much is due to breaking down DNA or how much of these endogenous chromophores are responsible for it. So we're getting a net effect when we do the applications, which I'll talk about in a minute. Now, we talked about efficiency again, and so what you see is a chart here that gives you sort of the range of doses that it takes to reach 99% kill. And what you see is viruses are very sensitive. They're very small. They don't have a lot of protective mechanisms, so a lower dose to reach a 99% kill rate, well, inactivation rate, I should say, for the viruses. Then bacteria are larger, and fungi are larger still typically. Now, you'll notice there's a lot of overlap, though, so you can't just go by size alone. You have to begin, again, to think about what is the pathogen I'm really trying to address. But broadly speaking, viruses are the most sensitive to ultraviolet, and fungi are the least sensitive. Now, what you see here is a breakdown, the different colors of how fungi are distributed and how bacteria and viruses are distributed in terms of dose. So you can see there is a range. Even some viruses are less susceptible to ultraviolet radiation than some bacteria, for example. Now, I think this is an illustrative example of why they differ. Powdery mildew, and I'm not telling you anything you don't know, is white in characteristic. And if you look at downy mildew, the spores tend to be heavily melanized, which means that melanin is a protective sunscreen against ultraviolet. Now remember, there's no UVC in the sunlight, but there's plenty of UVA and UVB. So melanin is a very common human skin, a way of protecting against solar radiation. It's interesting, I read a paper recently that the bacteria and fungi found after Chernobyl, almost all of them are heavily melanized. So despite the diversity you might find outside that zone, melanin is a very powerful way of protecting against ultraviolet or light in general. What that means is, from that previous graph, that you're going to have a higher dose to deal with downy mildew than you would for powdery mildew. So here is some results that we've gotten. So if you look at cucumber, downy mildew, heavily melanized, we were up to, and you don't have no sense of scale, but this is a high dose, 480 joules per meter squared, and we still were not very effective at reducing the prevalence of downy mildew in cucumbers. However, for powdery mildew, half of that was extremely effective. So again, it depends on what you're really trying to mitigate. Now, something like a bacterium, remember that scale, angular leaf spot, I mean, we completely wiped out angular leaf spot when we were able to apply it. And I'll show you a picture of that in a few minutes. About it really is day and night. So again, it depends on what you're trying to treat. It's great for bacteria. It's great for powdery mildew, not so good for anything that's melanized like downy mildew. Now, all of our applications are done at night, and you say, why is that? Well, it's not because we are it's called late chronotypes, which means that we're night owls when it comes. It has to do with the repair mechanisms available to many, if not all, plants, and it's activated by blue light. Now, blue light's in the visible spectrum. It's a longer wavelength, and so what it does is at night, there is no blue light if all you're treating it is with ultraviolet. So what you're doing is, unlike natural sunlight, where it has blue and ultraviolet, you're removing that repair mechanism, and it turns out to be extremely effective to apply it. Now, in practice, we try not to apply UV just before dawn because we have found that it takes if you apply it within about 4 hours of sunrise, you begin to reduce the effectiveness. So blue light will actually repair even immediately after the dose, at least up to 4 hours. So the idea is that we're always trying to apply it in the first part of the evening after sundown. So that's why we call these the dragon. I'm not quite sure how we did it. I thought it was dragon as if you're dragging the trailer behind, but our colleagues interpreted it as more of a beast rather than a trailer. But anyway, you can see on the right-hand side one of our squash dragons and how it's being applied in a nighttime application. So you take a 3-point hitch, pull it by the tractor, and at the right speed, then you can be effective to delivering the prescribed dose. Now, we have been using what are termed low-pressure discharge UVC lamps, and they look like a conventional fluorescent lamp. You see that in the lower corner. But you'll see the emission spectrum. What you do is you ionize mercury vapor, and the maximum is emission in these lamps is 254 nanometers. Now, if you remember, it was about 260 was about the peak. So this is a little bit less efficient, probably 80% as efficient as what you would get at 260, but it is still very much near the peak of the sensitivity of nucleic acids. So this has been a very effective one. And I must say that this goes back at least 100 years. In treating things like tuberculosis, airborne tuberculosis. It's used in a lot of shelters. It's used in purification of water. So there's a lot of applications where these low-pressure discharge lamps have been used successfully, again, over many years. Very rarely, if not at all, until we began in our collaboration with Cornell University to begin to look at this at controlling plant pathogens. So this is one of our first ones. Now you'll notice that this is for strawberry, and we only used 85 joules per meter squared. We weren't quite sure what was going to happen. We were just really exploring it. Now, strawberry is an alpine plant, and therefore we believe, because of the thinner atmosphere, it's probably very resistant to ultraviolet, damage to the leaves and so on. So we can have a high dose, or you can have a relatively high dose and still be effective. But this is working with a low dose. Now, what I show here is that the control, which means we applied nothing in this field trial, we took the standard chemistry that was being applied, and you can see it's effective, but UVC at 85 joules per meter squared was more effective than the conventional fungicide that was being used in this particular farm down in Florida. So we applied weekly. Again, we started wondering how often do we do it, and we found that weekly was satisfactory for this particular plant, and it was highly cost effective. So this particular grower has now gone through making his own 2 or 3 generations of the dragon, and many more rows than what we're having here, and is working with us hopefully in the future to begin to transition to a new light source, which I'll talk about in a minute. Now this is, you can see on these 2 comparative graphs, the big difference between treating zucchini with UVC weekly compared to not weekly. It really is a night and day change. And you can see here on the histogram on the left how effective it is. Now this is for 240 joules per meter squared, so we needed a higher level, but it's very, very effective without any phototoxicity on zucchini for this particular application. And I believe that's the first time anybody's looked particularly at bacterial infections on plants. Now, it's not all rosy. So here's an experiment we did with, I mentioned again, downy mildew was a very difficult one. We had 480, as I mentioned before, joules per meter squared. UV alone made a dent in it, but the weekly fungicide that was used was more effective for treating downy mildew. But what we have found time and time again, when you combine the traditional fungicide with UV, you always do better than either alone. And that's because they have different modes of operation. So this might be something not necessary to replace fungicides, but used as an adjunct with fungicides for maximum control. Now, people are always concerned about safety. And on the lower left, what you see is it's really about penetration into the skin. And you can see from this is solar radiation. There is no UVC in sunlight, but the penetration depth is much shallower. And it is true, if you're exposed to UVC, you will get rashes or what's called photokeratitis of the cornea, very painful, repairs in a couple of days, but why go through that? So we have been very conscious about safety issues and have never had a problem. We put curtains on the dragons, and we wear protective gear when necessary. But glass is opaque to UV-C, and so as long as you stay in the cab while it's being operated, you will not have a problem. And again, once a little caution like this is much better than it would be for worker safety than many fungicides. Now, the problem with low pressure discharge, I said they've been around almost 100 years now, is that they have, they can break. They have a quartz glass tube. Remember I said it doesn't pass through glass, but through quartz it will. The problem is it's temperature sensitive. So if you're doing something at night and the temperature drops, your efficiency, as you can see in the upper right, considerably goes down, because they're all designed by collusion amongst all the light bulb manufacturers to maximize at room temperature, which is they optimize about 77 degrees. But this is the way it's arrayed inside the dragons that we've been pulling, and it is very effective. But they have banned fluorescent lamps. Those of you that have them, you're going to have a difficult time getting them. And now today, they're even more expensive than they used to be. And the European Union is going to ban these totally, because mercury is considered to be one of those environmental hazards. So if it breaks, you will have mercury a lot less than it was in the 70s, for example, but nevertheless. So this is the reason why these are going to be banned. Now, the way the world is moving, and if you went to buy a light bulb at Home Depot or something, you would get an LED source, a light emitting diode, which is solid state. And you can see the size of them here in the picture down at the left. They're very tiny, solid state, they don't break. And the rate at which they're improving their efficiency, which means when is it going to be really cost effective to use LEDs, we see the crossover point in about 5 years where the price or the cost will be comparable to what it is for low pressure discharge now. Nevertheless, we already have projects going on right now with LEDs, working with manufacturers in Japan and Germany specifically to design LED systems in anticipation of their efficiency getting better. And here's a picture of one of the what we call LED modules that we've been using to do that. And this summer we'll be using devices like this in an application to control powdery mildew and squash. So maybe next year we can give you the results of those. But once again, you do the dose. It really doesn't matter whether it comes from low pressure discharge or from LED, as long as you take into account the efficiency. These peaks are about, I think, 280 nanometers for practical reasons. So they're comparable to what the 254 is on the other side of the efficiency curve, but they don't contain mercury. They're solid state. And this is the way it's going to go, not just for agriculture applications, but nearly all water treatment now is moving to LED, even though it's more expensive because of the risk of mercury into the water system. So this is the future. This is the way it's going to be, at least within 5 years, if not sooner, for water disinfection and hopefully for agriculture applications. Now, most of you are interested in, I think, controlled agriculture. And I just want to point out, here's the solar spectrum, which we rely on the far right. And then down below shows you the various transmission of what appears to us to be clear, but depending on the material, it can transmit more or less than you're going to get from a traditional glass covering. So if UV is going to be part of your recipe in a greenhouse, you have to make sure that the glazing admits ultraviolet, and you can get this technical information if you ask for it. But it's not at night. By definition, you're going to have ultraviolet during the day. But one of the things that we're exploring, and we're very optimistic, is using one of these secondary mechanisms, which is called exogenous, or outside the cell. And this particular material we're working with, very inexpensive, is titanium dioxide, and it combines with water and ultraviolet radiation to be highly effective. Within 10 minutes, you can probably kill, again, 99 or 99.9% of the pathogens when that mixture is applied. So this, what we have done is we've looked at one of the primary pathogens that affect apples, which is fire blight, and it's a bacterium. Erwinia is the target. And right now, most of the practice deals with antibiotics. But what we were able to show is that in the first trials we did, it was very effective against fire blight. And in the latest trials, which we just completed, it was shown to be more effective than antibiotics in controlling fire blight in this particular application. So an idea would be to, for example, in a greenhouse application, to take UV, and you can see it doesn't have to be UVC because the spectral absorption extends down almost to the visible region, so down into the 300 region, so UVA, UVB. And these LEDs are very efficient right now and cost effective, more than the UVC. But by supplemental lighting, like you see here, and then spraying, we use a back sprayer, and we just suspend the titanium dioxide in water. and we're getting significant reduction in the plant pathogens that we've been looking for, like Erwinia in this particular case. So this is brand new, and it's something that I think you want to keep on your radar screen, because I think it might be a way to really be effective and perhaps move away from the dominant use of chemistry to control pathogens. So thank you, everybody. I think I finished on time, with enough time to have questions, if there are any.
Yeah, that was wonderful. Thank you so much, Mark.
Sure.
If people have questions, they can put them in the chat, or we also may have time even to take an actual verbal one if somebody wanted to raise their hand or something.
I have a question. This is Rich. Just with the photo repair that you get with the blue light. So are you trying to prevent, do you not want the fungus to repair, or do you don't want the plants to repair? It's the fungus.
Right.
Yes. Yeah. Okay. That's what I thought.
That's right. That's why we do it at night. The efficiency goes way up if you do it at night, because you defeated that. I just want to say that you can send me an email if you have any questions, if that's easier for you. But
so one question was, how could this be used in ornamental mats outside? Because you can't drive a tractor on mats. And Sarah, you want to just explain what you mean by oriental mats? You can just unmute yourself.
Audio? Video?
Yep, good.
So I guess what I'm trying to ask is, in my practice, I grow most of my plants, they're ornamentals, outside on the big black woven mats. And I can't drive a tractor on the mat to use the UVC Dragon, but is there a way I can use some handheld method to cover my rows?
I have two answers. One is, yes, we have been working with Sarah Petherbridge at Cornell University, and she has, I forget what they call it exactly, but it's a death wagon. It basically is a device that you take and hand push over it. You have to get it the right duration, but it's been very effective at controlling beets. I will add beets in particular, though. Beets, there seems to be no free lunch, is that the more effective the radiation is, the more phototoxicity you get in beets, unlike something like strawberries. So I just want to caution you that it isn't a free lunch necessarily. It depends on the plant. And beets are particularly apparently sensitive to ultraviolet. The second answer I would give you is that you could adapt the technique we use to control Erwinia fire blight by using titanium dioxide outdoors during the day. And you could try that and see if you can, in fact, work that into your regime. It tends to be very effective. EPA says it's inert. I don't think USDA has made a ruling on it, but the Federal Drug Administration says you can eat it, and in fact you do. It's on pretty much, it's a whitener in all toothpaste, even powdered donuts. So we're consuming titanium dioxide in our diet practically every day, but it's not been formally approved as a treatment for pathogens in plants yet. And I don't quite know what path to take on that, but the fact that we can stop the use of antibiotics on apples seems to be a real plus. So hopefully that will be recognized in the near future.
Great. I need to go on to a couple more questions. Thank you, Sarah. I guess one quick thing, just to make sure everybody knows, UVC is an approved organic, or I don't know if accepted. It's an acceptable treatment for organic production, just so you know. So this is a question from Trish Ayers. Where does a person get a UVC LED light that can be suspended in a high tunnel? Is this what is recommended? Also, I am a small no-till farmer. Is there another way to apply to the fields?
Well, again, going the opposite, what explanation is that I recommend considering in your own trials to try titanium dioxide during the day with a back sprayer and suspended in water, and we can tell you what the concentrations need to be.
Huh.
I think you'll find it extremely effective because it is used in many applications, but I'm not aware of any that have been done in agriculture.
Okay.
But to answer
sorry.
the first one, I don't think you're ready for LEDs in your high tunnels yet. They're not as efficient as a low pressure discharge. So if you're willing to tolerate obsolescence in 5 years, I would recommend you think about that. And then make a measurement of how much radiation is, and that'll tell you what the dose is, how long it has to be operated. I'll just make a quick comment that they've done this in some of the greenhouses at Cornell, and we helped them set that up. And it's been very effective at keeping disease pressure down by having it on for 5 or 10 minutes at night. So it can be quite effective, but those are all low pressure discharge sources.
Okay. I'm not going to be able to, all these questions aren't going to get asked, but there's one important one. One is, how is titanium dioxide applied? This is from Gary. Would inhalation be a more risky exposure than oral exposure?
No, that's a good point. There was a study done with mice, and they did find that in high concentrations, titanium dioxide did lead to respiratory problems in the mice. That study was then tried to be replicated in Japan. It was not replicated. So the European Union, I believe, has reversed their position on that. So I'm not saying it's safe. I'm just saying the evidence is not compelling epidemiologically or in animal studies that inhalation is going to be a problem for human safety. But I'm a belt and suspenders sort of guy. I would still wear a mask. We do when we apply it. So why take a chance is sort of my view.
Okay, just quick, there's a few more questions here. We won't get to them all, but we will have all these questions and hopefully get to them at a later date if necessary. Phytotoxicity of titanium dioxide. How often do you apply it? That's from Rob.
Well, again, we're beginning our studies. I know of we have nothing so far in apples that would indicate any phototoxicity with titanium dioxide. I'm not sure that's true in all plants, but we've not seen anything related to it. We are beginning studies, so we were doing it weekly. It tends to survive mist, light rain. It does not survive on the plant for heavy rain, and it needs another application if you want to do it, but it's remarkably adhesive to the leaves that you don't have to apply it every day. It seems weekly was very effective for Erwinia.
Okay. We don't have time for any more of the questions, but I do want to mention, I don't know what the pesticide regulations might say about using titanium dioxide on your plants. And I don't know if there's anybody here that can speak to that, but you can't just apply any old thing to your plants legally. And so it might be something you would need to check with your state pesticide applicator people, just to make sure before you do it.
Yeah, I hope
moving along.
Okay.
I hope I made that clear. I was not making any claims that it was. I thought.
Yep.
I
Good. Yep. Okay. Thank you. All right. Here we go. Thank you so much, Mark. I think you stimulated all sorts of interest and questions and warrants more research. Thanks. Okay.